So we went through the reproduction outline, and I talked about, what was there, we could say for the sake of completeness, versus what had been covered, like the reproductive entomology, what was actually biology 111, like gametogenesis, and how the attempt was to make the outline all-inclusive, but the specific parts that we will cover, because they have not been covered, are sex determination, ejaculation, and the molecular events of fertilization. You probably are familiar with sex determination just from meiosis and genetics back in biology. But if you consider a female, 44XX, that means that all of her eggs will be 22X. There's no other possibility unless we get into Turner syndrome and abnormal situations, which is not the topic today. Males are 44XY, so through the process of spermatogenesis, you can produce X-bearing sperm or Y-bearing sperm. And so there's your sex determination right there. It's determined by the sperm. It's determined by the father. The ridiculous books you can buy about what to eat, what not to eat, what position to have coitus in, they're just that. They're ridiculous. So if the egg comes together with an X-bearing sperm, when the diploid chromosome number is restored, you have a female. On the other hand, if it's a Y-bearing sperm that fertilizes the egg, then you have a male. So whether we want to say sex is determined by the father or sex is determined by the sperm, I mean, you're saying it the same thing either way. All right, the next topic that I said we needed to touch on is ejaculation. You know exactly what is in the ejaculatory fluid, or exactly what is in semen. A very small component of it is sperm. About 1% of the semen is sperm. It might be 2%, it might be 1.5%. As with all of the numbers, I like to ballpark things for you to make it easy to keep up with. The majority of the semen comes from the seminal vesicles. One of the most important things that the seminal vesicles produce are proteins. And draw from biochemistry, that proteins are the most diverse group, the most abundant group of biological molecules. There just aren't that many carbs or sugars in living creatures. There just aren't that many fats.
Somebody staring at you or something?
The nursing students, they're all excited. Most of them finish tomorrow. They're just on cloud nine.
Yeah.
A lot of them are burning their white scrubs. I hate those things.
Yeah, who chose white?
I don't know. Anyway, so proteins, most abundant, most diverse of all of the biological molecules. Semen has thousands and thousands of proteins in it. We don't even know what they do. I mean, the bulk of what we'll talk about when we get to Roman numeral 8, the sequence of events leading to fertilization, we'll talk about a few of the proteins, but nobody knows what they do. We just know what a small handful of them do. The seminal vesicles produce prostaglandins, in addition to the proteins. They produce an alkaline fluid to help neutralize the acidity of the female reproductive tract. They produce fructose. About 35% of the fluid of the semen comes from the prostate gland. We'll look at all of these glands in a minute, but keep in mind you've seen the prostate gland because we've seen the urethra and how when it leaves the urinary bladder it courses through the prostate gland and is called the prostatic urethra. The prostate gland produces several things, but among them are antibodies. It produces an alkaline fluid. It produces a liquefying agent. Imagine if the sperm were to, or if the semen were to coagulate, the sperm would never reach the egg. And then finally, a little bit of the semen comes from the bulbourethral or Cowper's gland, maybe 5%. I say a little bit because what the bulbourethral fluid truly is, it's a lubricating fluid. It's known as pre-ejaculatory fluid. So the idea is that the lubricating fluid, the pre-ejaculatory fluid, is released prior to coitus to aid in penetration. Now the only gland here that females have something similar to are Bartholin's glands. Okay, so these, again, are in females, and they produce lubricating fluid for the same purpose, to assist in penetration. Glance at these structures. We've seen the prostate gland numerous times, just inferior to the urinary bladder. Seminal vesicles are here. This duct, and you can see this in the model in the lab, is called the ejaculatory duct. It carries the products from the seminal vesicle to the prostatic urethra. And this little gland right here, that's Cowper's gland, or the bulbourethral gland. No, I'm not adding these to your practical. I'm just showing it to you. All right, so the bulk of what we want to talk about this morning is Roman numeral 8, the sequence of events leading to mammalian fertilization. What I want you to keep in mind is fertilization is truly and literally the coming together of the two haploid nuclei. So the male and the female have both produced gametes. You know all about that from Biology 111. I told you the other day, at this point, I'm not going to test you on the events of meiosis the way you were tested in Biology 111, but a superficial, big picture type of understanding is what you want with regard to meiosis. And of course, the fancy name is gametogenesis. But literally, fertilization is the coming together of the two haploid nuclei. Now, these pictures are not to scale. If I draw the picture to scale, if that's the egg, here's the sperm. You can't see it. I can't see it. So we can't draw things to scale. So somehow we've got to get those two haploid nuclei together. Think about chromosomes again. Go back to biology. You have 46 chromosomes, or 23 pairs. What do we say about the pairs? They're homologous pairs, so there's two number 1s, two number 2s, two number 21s, two number 22s, and two sex chromosomes, either two X's or an X and a Y. So we've got to get the two haploid nuclei together. Literally, what is going to happen is the two haploid nuclei are ultimately going to fuse. So now we could say the male chromosomes and the female chromosomes are encased with one membrane, but the homologous pairs aren't paired up. It's kind of like we have the boy chromosomes over here and the girl chromosomes over here. They have to come together, homologous pairs. And so finally, what we will end up with is a single diploid nucleus, and we'll have a zygote, or a one-cell embryo. So that's like reading the last page of the book almost. What we want to look at is, how does this happen at the molecular level? And I promise you, when I finish telling you, you'll be amazed that anyone has ever gotten pregnant.
Why are some people more fertile than others? Seriously, like why does that happen?
There's not one single reason for that. It could be the number of sperm that the male produces, the motility of the sperm. It could be something with the tissue in the female reproductive tract. Remember the whole, let's see, I've got a quick picture here. Remember the whole business with where fertilization occurs in the simple ciliated columnar epithelial tissue. So the sperm are ejaculated right at the cervix. Fertilization occurs way out here. So you get into motility of the gametes. It could be something to do with the endometrium. There's not one answer to that. All right, so first thing we'll do is now give yourself a clean sheet of paper turned sideways. This is one of those ginormous pictures. Maybe take two sheets and duct tape them together. So we're going to put the egg here, put the sperm over here. I'm going to use this area for what I call sidebars. You're all familiar with that. And then once we get the anatomy of the gametes down, we'll get into how it actually happens. So there's the beginning of our egg. Has a cell membrane, just like any cell. Superficial to the cell membrane, there's a glycoprotein layer. It's called the zona pellucida. It's actually made of 3 glycoproteins. One is called ZP2. It's the major component of the zona pellucida. It's the structural component of the zona pellucida. Another of the glycoproteins is called ZP3. Let's pay careful attention to our drawing. Initially, I labeled the red just the zona pellucida. That was before I pointed out that the zona pellucida is made of 3 glycoproteins. I'm color coding the key over here. So let's go back now and specify that that is actually ZP2, and that's not a subscript, it's a 2. The green is ZP3. ZP3 is the egg's primary sperm binding protein. Get that written down and think about exactly what it means. It's the eggs, okay, so showing possession, it belongs to the egg. It's a binding protein. What's it going to bind? What's going to bind it? The sperm, okay? And is it going to be the first, second, or third protein to bind the sperm? The first, that's why it's primary. What would you call the protein on the sperm that binds this protein?
Primary sperm binding protein.
First word,
Sperm.
sperms, primary.
Egg binding protein.
Okay, so let that make sense, okay? It does make sense. The third protein of the zona pellucida is deeper in the zona pellucida. And it's ZP1. And it is the egg's secondary sperm binding protein. So do the same thing we did for the green one. What are you going to call the protein on the sperm that binds this purple protein on the egg? Sperm secondary egg binding protein. Okay, very good. Outside of the zona pellucida, there's another layer called the corona radiata. Worked well. I can tell that's one of those markers that's not going to erase very well. There we go. Corona radiata. And outside of the corona radiata, there's another layer called the cumulus oophorus. As you're drawing this, as you're looking at this, remember that what we're trying to do here is get two haploid nuclei to come together. Look at all these barriers, look at all these layers. Okay, a lot of times we lump the corona radiata and the cumulus oophorus together and just refer to them as the cumulus. Okay, so there's our egg. Now the sperm. There's a vesicle in the sperm called the acrosome. There's nothing complicated about it, except students find themselves confused when they watch it react, because it's not shaped like a biology vesicle. So let's clear the sidebar area to make room for more sidebars. So when I say students, it's not complicated, but students struggle with it because it's not like a biology vesicle. In other words, in biology, what did you see vesicles do? Or even this semester, with the release of acetylcholine at the synapse, what did you see happen? The vesicle moves over and fuses with the cell membrane, and then you get a rearrangement in the cell membrane, and whatever's in the vesicle is released, right? So this is no different. That's what I'm telling you. It's just that it looks different because it's not round. It's shaped like a horseshoe, but it's going to do that very same thing. Okay, we want to color code things here. So primary is green. These green ones are the, tell me again, what are they?
Primaries.
I know you missed a word. Sperms, primary, what?
Egg binding protein.
And they don't have some other name like ZP whatever. That's just what they are, the sperm's primary egg binding protein. Okay, the secondary binding protein, using purple for that. It's actually inside the acrosome. Sperm's secondary egg binding protein. Well, there's a nucleus in the sperm, a haploid nucleus. And right about now, somebody often asks, or at least wants to ask, why are all these binding proteins there? And I mean, it's clearly it's going to be like a hormone receptor event. An enzyme substrate, a lock and key. Why do we need these binding proteins? Well, as I've said so many times, we can check the owner's manual. And when we open the owner's manual, we find that it's blank. So you text God, you text Darwin. We'll see what they say. Presumably the binding proteins are there to help maintain species specificity. But last time I checked, in my neighborhood, we don't have a problem with dogs and cats mating. When I was on the Great Safari, I didn't see zebras and giraffes mating. But when we think of it that way, we forget that most animals don't have sexual intercourse. Most animals live in the ocean. Jellyfish ejaculates into the open sea, we could say. How do we make sure that jellyfish sperm bind jellyfish eggs and not sea urchin eggs? And how many species of sea urchins and jellyfish and frogs and whatever are there? So perhaps that's why we have these receptors. Species specificity. There's a third protein here. Now before I, what's the green protein on the sperm going to bind? The green protein on the egg, right? So while it's a mouthful, it's quite simple at this point. The sperm's primary egg binding protein has to bind the egg's primary sperm binding protein. But it can't right now. Because there's another protein called the inhibitor. Okay, that's pretty much what we need to tell the story now. Put it in steps 1 through 8. Step 1 is called capacitation, as you see, looking at the notes. This takes us to one of those famous stories in biology. I alluded to this when I talked about complement fixation. I told you that was one of those famous stories where we think we understand how something works, let's go into the lab and try to do it. And on that day, I reminded you that people used to believe that inside the sperm lived a miniature person. The egg nourished said person until he or she was big enough to live outside the womb. Remember? That famous picture is still in a lot of the general biology books, right at the beginning of the genetics type discussion, DNA type discussion. So when we realized that, that's not how things worked, we went into the lab and we tried to create embryos. So they ejaculated a mouse and they harvested the eggs from a female mouse. They put them in a test tube, shake, 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 put the test tube down, went home, went to bed, slept, came back the next morning, no embryos. Tried it again, didn't work. Tried it again, didn't work. Tried it again, didn't work. Tinkered with the fluid. Should we modify the salt concentration? Should we do it in plasma instead of saline? Didn't work, didn't work, didn't work, didn't work. And somebody, last name was Lilly, this was about 1912, and we played checkers that day. It was a cold summer in Telluride, I remember specifically. Lilly said, let's incubate the sperm in a female reproductive tract before we combine the sperm with the eggs. So they did. They ejaculated the mouse, put the sperm in a female reproductive tract, went home, went to bed, came back, washed the sperm out of the female reproductive tract, mixed the sperm with the eggs in a test tube, and they got embryos. And Lilly said, and I can't quote anymore, but basically what Lilly said was, apparently during the overnight incubation period, the spermatozoa were endowed with the capacity to fertilize the eggs. Endowed with the capacity to fertilize the eggs. And the term capacitation was born. And for decades and decades and decades, nobody knew what happened during capacitation. They just figured that whatever happens, happens. Sperm's released at the cervix, it's got to get to the egg. It just happens. For decades and decades and decades, researchers maintained colonies of animals to serve as incubators. Capacitation was very quickly, they learned it was not species specific. So you didn't need to capacitate in humans to work with human gametes. Didn't need to capacitate in rabbits to work with, okay. So they use mice primarily because they're small, they don't eat much, and they're cheap. So they maintain colonies to serve as capacitators. After decades and decades and decades, somebody finally created a capacitating fluid. So you could now soak the sperm in the fluid and it would become capacitated. They still had no idea what was happening during capacitation. People studied the migration of the phospholipids and the proteins through the membrane. And they had no idea. Anyway, it was finally revealed that the primary event of capacitation is the removal of the inhibitor. So, I'm going to have 3 sperm over here. And I know I didn't draw number 3 yet, and I don't want to draw number 3 yet. Actually, I don't even want to draw number 2, really. So sperm number 1 looks just like the sperm on your left, just smaller and sloppy. So sperm number 1, again, just like the one on the left, is non-capacitated. Sperm number 2 is capacitated. What do you know about, that you have studied in this room and at home, that you can use proteins to produce, especially when you introduce those proteins into an animal of another species? Antibodies. I don't know how many of you have taken microbiology. Some of you have, some of you probably are, all of you probably will. But in microbiology they do, and I think it's more of a demonstration lab than you do it yourself, but they use an immunofluorescence microscope. And so you can tag antibodies to glow different colors under the immunofluorescence microscope. And it's a really cool demonstration. I know that Carla Ballou does it, and Montgomery does it. I'm not sure if everybody does it. But imagine making antibodies against all 3 of these proteins, and they glow different colors. And so you have sperm, and you don't know what state it's in. You throw all 3 antibodies in there with the sperm. If the sperm is non-capacitated, the antibody made against the inhibitor will bind the sperm. So the head of the sperm, so to speak, will glow that color. If the sperm's capacitated, then the antibody made against the primary binding protein will bind the sperm, and the anterior aspect of the sperm would glow that color. Pretty cool use of something. All right, so the sperm is capacitated. Now we assume that in normal in vivo fertilization, the time from ejaculation to the sperm arriving at the egg, the inhibitor falls off. Next thing we have to do is get through the cumulus, step 2. Today we don't yet know exactly how the sperm gets through the cumulus. We used to think, we know it's not this way anymore, in spite of the fact that it's stated this way in some of the books. We used to think that the acrosome released something that digested a path through the cumulus, but we know that's not true. The acrosome is still intact, as it is in sperm number 2 over there. A guy in Japan studied the movement of the sperm through the cumulus, and he noted that as the sperm got closer to the cumulus and started boring through it, the flagella beat faster and faster and faster. And he wrote a paper, loosely speaking, it was called, and this is one you definitely want to write down to throw out at your next dinner party with neighbors who are accountants and engineers. Hyperactivated flagellar motility. That was the name of the paper, something like in murine spermatozoa, which means in mouse sperm. Hyperactivated flagellar motility. Now you guys know about the scientific method and stuff, and so you study something, you write a paper, you get more money to study it further, you get more, and that's the way the world goes around. You publish your results, you get another grant, you do more work, you publish your results, you get another grant, you do more work. This guy got $1 million or so for that paper about hyperactivated flagellar motility. So we say that the movement through the cumulus is mechanical. And I'm sure eventually we'll learn more about it. After the sperm gets through the cumulus, there's a loose association between the gametes. I'm using the word loose versus tight. And that's called attachment. I think that's step 3. It is step 3 on your notes. Now, if you look at step 4, and I want you to somewhere make a little chart, not here, but I want you to make a little chart and differentiate between binding and attachment. So attachment is a loose association between the gametes. Binding is a tight association between the gametes. Attachment loose, binding tight. Attachment is not species specific.
What is not species specific?
Attachment is not species specific. Binding is species specific. Now it's your turn. You guys tell me. If attachment's not species specific, but binding is, what makes binding species specific? What proteins? The binding proteins. That's what you meant, right? The binding proteins are what make binding species specific. So we've got attachment loose, binding tight. Attachment is not species specific. Binding is species specific. Attachment does not utilize binding proteins. Binding does utilize binding proteins. Step 4, you can read that one straight off the notes. It's called primary binding. So using language that you've already used, would you define primary binding as the binding between the sperms, come on,
Egg
Mr. Ward, the sperm's primary egg binding protein binds the egg's primary sperm binding protein. I know it's a mouthful, but it's civil at this point, right? So that's primary binding. Now here's the cool thing about primary binding. Okay, now the gametes are connected, physically connected, it's tight, it's species specific, all of that. But here's the thing, and you can read this on your notes as well. Primary binding induces the acrosome reaction. Primary binding induces the acrosome reaction. Now you guys have many more pages because I keep needing more space. I used to have a board over here and I miss it very much. Did I tell you guys the story about the board over here?
I fell off and hit someone in the head.
At Centennial campus, it turned over, hit a girl in the head, broke her glasses, cut her face, and so they just removed this one before somebody got hurt. All right, so acrosome reaction. The acrosome reaction is that exocytotic event. Now we're going to, even though I already reminded you, I'm going to remind you again. When we think about vesicles and exocytosis, we're used to vesicles being round because that's how it always is in biology. It's the way it was here with the release of acetylcholine. So we see the vesicle fuse with the membrane, then we see the actual exocytosis and the vesicular contents are released. That's exactly what's going to happen, except look at the shape of our cell, look at the shape of the vesicle. So if this acrosome moves forward, and fuses with the anterior aspect of the sperm, those secondary binding proteins are in there. The primary binding proteins are out here. When those membranes fuse, I need everybody to be watching this. Now which antibody will bind the sperm? The one made against the secondary. And the sperm would glow whatever color that antibody was tagged with. Here's all that happened. This is all that just happened. A vesicle fused with the membrane, rearrangement in the membrane, vesicular contents released. Just like the release of acetylcholine at the synapse, except the vesicle's not round. It's not complicated, but it bothers people. So hopefully I've made it clear to you. So sperm number 3 is acrosome reacted. Now what gets released from the acrosome, okay, I told you over here it's a vesicle. It contains an enzyme called acrosin. So primary binding holds the gametes together initially, but also induces the acrosome reaction. And let's just make sure your notes say that. They do. Step 4, primary binding, the sperm to the zonas, EP3. Step 5, acrosome reaction, reduced by primary binding, releases enzymes. The enzyme is acrosin. Now, what happened to these primary binding proteins when the acrosome reaction occurred? They're gone, right? It's not a trick question, they're gone. So why do you suppose the secondary binding proteins are there? To bind the egg, secondary binding protein, to hold the sperm in place. If we didn't have the secondary binding proteins, the sperm might back out. So literally, after the acrosome reaction occurs, the secondary binding proteins are there. Now, how does the sperm get? I see the secondary binding proteins are on the inner aspect of the zona. Primary are on the outer. How's the sperm get from the outer part to the inner part?
Does the enzyme break down the glycoprotein?
Yes, exactly. That's what acrosin does. God, Stephanie, you're a genius. Seriously. Acrosin digests that Red Bull or whatever you're drinking. Acrosin digests the zona pellucida. Now I talked about how when we finish all this, you're going to be surprised anyone's ever gotten pregnant. Are you becoming that way? Is it not sort of a trip? So, primary binding, acrosome reaction. Secondary binding holds the sperm in place. All right, now let's clean all this up. New sheet of paper. We'll leave the egg there just in case you want to refer back to it.
So did we just talk about 6 then, secondary binding? It just means that it holds it in place?
Yes, as we continue all this digestive process. All right, so what number is next? 7?
Yeah.
And it's called what? Fusion of gametes.
Yes.
And that's what literally happens. Again, we can't draw things to scale because we wouldn't see them. Okay, so the sperm has gotten. That's kind of why I said let's not erase that. So the sperm has gotten all the way through the zona. That's actually called a penetration slit, and I think your notes say something about that. Yeah. Secondary binding holds spermatozoa in proper orientation for penetration. So there's got to hold it in just the right place there. All right, so fusion of gametes. So the cell membranes of the sperm and the egg have come together. Those cell membranes actually fuse. Keep in mind, scale, the sperm is much bigger in the picture than it would be in reality. But it does contribute to the cell membrane of the embryo. The flagella is beginning to disintegrate. Fusion of gametes plasma membranes. Can't you just hear the people arguing? Oh, the egg's so big, the sperm's so small, the cell membrane of the one cell embryo's got to be from the egg. Well, most of it is, but part of it is from the sperm. There's a haploid nucleus in the egg. There's a haploid nucleus in the sperm. Fusion of the plasma membranes is complete. But if you think about it, what do we have here? We have a cell kind of sort of thing. But does it have a nucleus? Not really. What's it got? It's got two haploid nuclei. Now they're precursors to the nucleus. And so they're called pronuclei. Now you look at your notes, you'll see that the next step is called decondensation of pronuclei. Pronuclei, precursors to the nucleus. So in that cell, the two nuclei are going to fuse. Now remember, the first thing we did was we read the last page of the book. So we've actually already seen this. This is where I started. What is fertilization? Fertilization is the coming together of the two haploid nuclei. We had all these barriers, all this bizarre stuff that had to happen to get the two haploid nuclei in here. Now there's a fascinating, and I won't go into it, but there's a fascinating in vitro technique where they actually introduce the sperm nucleus into the egg with a micropipette. So you skip all this stuff. If it doesn't work for you, you just skip it all.
That's what in vitro fertilization.
It's called EPSI. It's a type of in vitro fertilization. Okay, so a couple more pictures to finish this up. The pronuclei are fusing. God, we've almost got a nucleus. It's so close. But again, guys, you've got to go back to biology, DNA, genetics, cell division, all of that. Homologous pairs of chromosomes. We've got 23 homologous pairs. So at this point, as I said 45 minutes ago or so, We've got the male chromosomes on one side, the female chromosomes on the other side. The homologous pairs have to pair up. And once the homologous pairs pair up, then we have an actual diploid nucleus, and we have a one-cell embryo, which is called a zygote. Again, sort of amazing that anyone has ever gotten pregnant. If you're ever, talking with a patient or a friend, it doesn't have to be something associated with your job, and they're having, trouble getting pregnant, think about all this stuff. You don't have to get a whiteboard and draw it all out. You guys can imagine when my son asked me, he was like 3, Dad, where do babies come from? Well, we got in the car and drove to the school, and I set it down right there and proceeded to show it. We started with GNRH, of course. But just think about this stuff. Now, there's one more thing, one more step, and I don't really like to call it a step, and that's why I didn't give it a number. I gave it an asterisk at the bottom, because we don't know exactly where it goes in the sequence. We can say, well, this sort of seems like where it needs to go, but, that's not science, is it? This is what I think. That's not science. Anyway, it's called the cortical granule reaction, or the block to polyspermy. Now, polyspermy would be the egg being fertilized by more than one sperm. So, for example, a triploid embryo. Well, that's not going to be viable, so do your best to prevent that. And again, I'm glad I left the original picture up, because we need to add something to it.
What's the definition of polyspermy again? I'm sorry.
Definition of polyspermy?
Yeah.
Poly, many. So
Okay.
fertilizing the egg by more than one sperm. All right, so just inside the cell membrane of the egg are vesicles. They're called cortical granules. What are those vesicles going to do?
Unlock the formation of polysperm.
Release the protein. So, if we take an area like this,
Isn't that where like mitochondria is? Mitochondria from the mom and all of the stuff the baby eats?
there's one of the vesicles. With me?
Yeah.
Vesicle fuses with the membrane. Exocytosis of whatever's in the vesicle. Now, if the vesicles, they all fuse with the membrane and exocytotically release their contents. Stephanie, you said a protein. Yeah. I don't know the name of it. But the contents of the cortical granules cross-link the proteins in the zona pellucida so that acrosin can't digest it.
Say that one more time.
I'm going to, but I also want you to think about it, because you've heard me say so many times, we've got to minimize what you have to memorize, so we've got to make sense out of things. So, maltase digests maltose. But what if you changed the shape of maltose? Then maltase couldn't digest it. So, the enzyme that the cortical granules release changes the structure of the zona pellucida so that acrosin can't digest it. So what you wanted me to say one more time is that acrosin cross-links the proteins of the zona pellucida so that acrosin can no longer digest it. So change the structure of the zona pellucida so that Acrocin can no longer digest it.
Where does all that stuff? I don't know. Like all the stuff, like the mitochondria and the antibodies that the baby needs to get going and.
Well, the organelles are in the gametes, primarily the egg. And then the antibodies, remember we talked about with regard to blood, plasma proteins. Plasma proteins are going to cross the placenta. So the baby's born with some initial immunity, and then antibodies will continue to be received through the breast milk. But this cell that we have here, that cell has everything in there. And that one cell is so now we get mitosis. So 1, 2, 4, 8, 16. And initially we'll get a division into two subgroups. One subgroup will form the embryo. The other subgroup will work with the endometrium to form the placenta. And then the subgroup that's going to form the embryo will get subgroups there to give rise to the different types of tissue, and on and on and on. It is amazing. All right, you guys have with you the reviews for exam 3 and the final. So, if there's any last minute questions there, and of course I've asked you more than once to treat these review sheets as take-home final exams. Whether it's a simple definition, or whether it's a concept, or write it out longhand, or do it on the computer with Microsoft Word. What you're creating, of course, is your own study guide. But if you have something you've been struggling with on one of those, if you want to ask, that's fine. Give the class perspective by saying this is the exam 3 review sheet, item 4, whatever it is, just gives people perspective. I do want to remind everyone that the retake on the practical, for those of you that are utilizing the retake on the practical, it's Monday only. It's not Monday and Wednesday. It's Monday only. Yeah.
For the exam, since there's no practical afterward, would that be longer? Like, will
Nope.
we have longer?
Nope. You have the same period of time, but you have twice as many questions. Just like any exam. It's worth more points. So the final's worth 250 points. So an 80%, which of course is 80 out of 100, you multiply that by 2.5. 80, 160, so that'd be 200 out of 250. So this person would see 200 in D2L. But I'll have grades in the lab on Monday after exam 3. I'll have grades in the lab on Wednesday after the final. You know what you guys need to do? Let's send the attendance out again, because we don't want to do attendance next week. You'll be testing. So unless this is your last day, and you're not coming back next week to finish the class, go ahead and sign in for next Monday and Wednesday. Questions off the review sheet? Everybody's ready. We can just go ahead and take the finals.
We're ready.
Remember, while you're thinking about whether or not you have questions, just go back through everything. Hold on to this stuff, guys. One of my analogies in 201 is that AMP 1, AMP 2, you're learning how cars work or how refrigerators work, because you don't stand a chance of fixing them when they break if you don't know how they work. You can't go to school to become a refrigerator repair person without first learning how refrigerators work. You can't go to school and become an auto mechanic without first understanding how cars work. So AMP 1, AMP 2, you just finished all that, congratulations, you're awesome. Now you understand how they work, but you can't forget it, okay, you can't just purge it, dump it, because most of you now are going into patho to understand what happens when they don't work. You know, when there's a breakdown in homeostasis, what are you going to see? And then most of you from there are going into, well, how do you fix it? So, you know, hold on to this stuff, do it as necessary. Course-specific stuff. Remember how final grades are calculated. You don't calculate an average. I use total points. So there's no such thing as this 89.9 round to an A. There's no such thing as 89.9. It'd be 899 points, right? 900 points is an A, 800 points is a B. If you need to review the material in your syllabus, do all of that, do that. All right, last chance. Any questions? I know a lot of you just are focused on the lab right now. You just want to go get it done.
Do you have any, special ones for us?
Special.
Yeah. Can you throw us a bone?
Yeah. Essentially.
Charles Houston.
Okay. Go meet him over the weekend.
I have no special hints. I wish I did. I mean, I don't. I would say,
It's just rather daunting that like 350 points is coming next week.
I know.
So don't think of it like that.
How should I think of it?
It's just a test.
She's really good at it.
It's just a test like any other. I hear you. I know. I mean, I remember it always seemed like college was that way, because the last week you have the final exam, the lab practical, the term paper, the oral presentation, not all in the same class, but it all, you know, it's like half your grade comes down to the last week of school. Yeah. That's what it is. All right, I'll see half of you upstairs in the lab. I'll see all of you on Monday.